From Microbe TV, this is Immune, episode number 6, recorded on March 13th, 2018. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, and welcome back.
You have snow today?
We are getting a little bit of snow, and I think we're going to get a storm coming in. But it's been really nice here the past few days. Quite sunny, delightful.
Yeah, we have our third nor'easter in 10 days here.
Yeah, I saw that. We're supposed to get the tail end of that, but I think you guys down on the coast have beginning to get a lot more than we've been getting inland.
We have hardly any snow today, but I saw a movie from Massachusetts, and it's really snowing there. They're getting a lot of snow.
Wow.
And, you know, the funny thing is, last week we had, like, on Wednesday, I don't know, 10 or 12 inches, and Drew University, half hour away, they had 23 inches.
It's amazing how that short distance can make a big difference. Yeah. Here, it's even more dramatic because we in Ithaca, if you've ever been here, there's a lot of hills and valleys, and I wouldn't call them mountains. But if you're downtown, it could not be snowing at all. But if you come 5 miles, you could have 6-8 inches of snow. It's pretty amazing.
Also joining us from Worcester, Ohio, Steph Langel.
Hey there. Good to be here. It's also snowing in Ohio. Transferred. I think it was quite nice this morning. The sun was out, and then all of a sudden I looked out and it looked like a blizzard out there. So
it snows in Ohio, huh?
No, it does. It does. The more north you go, the more lake effect you get, and it gets worse. But I think I'm about an hour south of that.
What's the most snow you ever had around where you are?
Oh gosh, I mean, on average, I mean, we could get 10 inches with a good snowstorm. We could get up as high as what you were discussing. Brian Barker had in New Jersey. So it just kind of depends. But because of the lake effect, we can get a lot.
Well, it is March 13th, which is halfway through March. And that means that April is coming up.
Wow, time flies.
And we don't get much snow in April.
April showers bring Mayflowers, right?
Showers. I remember once when I was young, we had snow on Easter. That's about the only, I think Easter is in April. I don't pay much attention, but
yeah, I don't know. It might be in March this year. Either it's March or April. It's always it's at the end of that March, beginning of April month.
Well, we're heading into spring pretty soon.
That's right. Now, when did April showers bring cherry blossoms? Your favorite?
No, no, we don't have. Yeah, we don't have any. I don't go anywhere where there are cherry blossoms.
I know you hate them.
I don't particularly like them. No, it's funny.
Odd.
Well, we have some immunology today. This being immune, and it is my turn. So I, as usual, will hide in the refuge of a paper that has a virus in it, just so I have something that I can sidle up against that I know something about. And this one continues an arc that I had that I started, I guess, the last time I talked about a paper. What was that? Monoclonal antibodies against Zika, bispecific monoclonal antibodies, right?
Yep.
And this will continue that arc. And this is a paper in immunity. So there you go. I'm picking the right journals, and they just happen to have viruses in them. And this is called Infants Infected with Respiratory Syncytial Virus Generate Potent Neutralizing Antibodies That Lack Somatic Hypermutation. And those last two words got me to this paper because I thought it would be a good opportunity to explore that. And this comes from a company called Adimab in Lebanon, New Hampshire, and also Dartmouth School of Medicine, the Vaccine Research Center, which is on the campus of NIH, and that's it. I got them all. And the first authors, let's see, these authors contributed equally, are the first three authors, Eileen Goodwin, Morgan Gilman, and Daniel Rapp. And the lead contact is Laura Walker, who is from Adimab.
I wonder how far we're going to get someday with the co-contributing authorship. If it goes, you just keep going, 4, 5?
Yeah, as many as they want, right?
Right.
Yep.
Right.
I used to be a lot simpler, I have to say. You would take the first person, that would be person, the first author and the last author, and it was simple. But now you have to, I learned on TWIV because Kathy Spindler always corrects me when I don't get all the right people in. So we're going to talk about this a bit, but first let's chat a bit about first respiratory syncytial virus. Now, this is an important virus, the most important viral cause of pediatric lower respiratory tract illness in the world. The whole wide world.
Yep.
It's a really important virus. I know and I know everyone else who has had kids probably experienced RSV. One of our kids had a very serious infection, really worried about long-term sequelae.
Right.
This is a member of the paramyxovirus family, and that family also includes measles virus. So it's an enveloped virus that has a negative strand RNA genome. And the envelope, this is important for our discussion today, there are 3 different glycoproteins in the viral envelope. One is called G, and that is the protein that attaches to the cell receptor. There's another one called F. These are creative names, G glycoprotein, F fusion protein, and SH, which is a smaller protein that appears to make pores in membranes.
So why is it SH? Do we know?
Yeah. There is. I had it out, and I said, "Nah, I'm not going to bother telling you that." SH stands for.
Is it small something because it
small
is
hydrophobic? There you go.
Okay.
Thank you.
There you go.
I guess that's creative, right?
Well, it's better. Sometimes immunologists, we name things that are just a lot of numbers, really, and then it can get confusing. At least this has a relation to the function.
Yeah. Do we know the receptor? Do we know what G binds to?
Don't know. It doesn't pop into my head. I don't think we do know.
I would think it would be something in the lower respiratory tract because that would indicate where it's infecting and causing a problem.
Yeah, I'm not aware of it. Could be wrong.
I don't know.
Anyway, the important part here is also there's one serotype. A serotype is an old definition for you take a virus and you get infected and then you make antibodies that will then block infection with the same virus. And so there's no antigenic variation with respiratory syncytial virus that appears to impact immune responses. Now, numbers of cases globally, 34 million infections, that was 2005, in kids less than 5 years old.
Right.
Almost 3.5 million hospitalizations, upwards of 199,000 deaths. There's a range between 66,000 and 199,000. And these are probably underestimates because 99% of the cases of the deaths are in developing countries.
Mm-hmm.
So that's the key. If you're in a country like the U.S., you can get cared for and you'll survive. But if you are in a country with not a great healthcare system, you have a good chance of dying of this.
Now, since this is a virus, it's mostly supportive therapy, correct?
Well, there is some therapy that we can give kids monoclonal antibodies, right?
Uh-huh.
We'll come to that. But infection is very common in infancy. Up to 69% of infants are infected during the first year of life. Now, you might think, where do babies get infected? Well, all over the place, right?
Yeah.
Parents, if their parents bring them somewhere, if they go to a doctor's appointment, if they happen to be in the hospital for something else, they can get infected. Lots of places.
Childcare, I'm assuming.
Oh, yeah.
Well,
basically, all kids are infected by the age of 2. And that first infection, and you get multiple infections in your lifetime, the first one is usually symptomatic. And if you're infected in between the 6-month phase of your life, that's when you go to the hospital typically. And it also will infect beyond the first year and cause respiratory disease. Tends to be as you get reinfected, the infections get less and less severe. But it turns out that in adults, it's second to flu as a cause of medically significant respiratory tract disease. And if you're over 65, so I am 65, so it doesn't count, right? If you're
Not greater than or equal to, only greater than.
If you're over 65, you start to have immunocompromising issues, and that's when this virus can cause problems again.
Yeah.
And all of this means to indicate that we need a vaccine, and you would think we would have one because we've known about this virus since the 50s.
Right.
And I remember as a grad student learning about the very first respiratory syncytial virus vaccine developed. It was tested in 1966. This was a formalin inactivated vaccine. So it's non-infectious. The viruses are treated with formalin to inactivate them, so they're not infectious. Injected intramuscularly. So they did a trial in infants and children, and this was a disaster. I remember the fellow who did this was Robert Chanik. He was at NIH. He was one of the people involved. The kids who were immunized got more serious disease than those who were not. 80% of vaccine recipients had to be hospitalized. So the idea is you immunize a cohort of kids, and then you have a control that are not immunized. They go out in the world and they get challenged, they get infected. And 5% of the kids that were not vaccinated were hospitalized, and 80% of those who were. So vaccination enhanced the disease. And for years,
this worst case scenario.
You don't want that, right?
No.
And then, of course, that was the end of that vaccine. But people spent years trying to figure out what was going on. And
I can imagine that hindered development of a future vaccine. People were
Sure. I mean, you have to know what happened with this one before you can go forward, right?
Right.
So first of all, the antibodies that were induced by this vaccine, they bound virus, but they didn't block infectivity. So these kids who were immunized, the virus was able to replicate in them. But that doesn't explain it, because kids who aren't immunized, the virus can also replicate. And one of the reasons why this vaccine didn't stimulate very good antibodies, this was found out not too long ago. The vaccine, because it was formalin treated, it apparently destroyed ligands for the toll-like receptors that sense this virus. And as a consequence, the antibodies that were made weren't good. The other thing is that the vaccine induced more Th2 type CD4 cells than you would see in a natural infection. Now, Cindy, what would that do if this happens?
Yeah, so we have these different types of T cells, helper T cells. So there's Th1 cells and Th2 cells. And the Th2 cells have a tendency to drive more antiparasitic and allergic type of responses, where you produce a lot more mucus, you produce a lot more mast cell activation and things like this. So what that's actually going to do in the lung is possibly make it worse, right? Because the cytokines that are produced by those cells are designed to help flush out the mucosal surfaces. And so if you do that in the lung, what you're going to do is you're going to fill the lung up with mucus, which is going to make respiratory infection worse.
Right. So Saturday Night Live, did you want to say something?
No, I mean, that's true. And I think the other thing is that if you're driving a Th2 response and not a Th1 response, which would be cytotoxic T lymphocytes, and these are the ones that are really critical for clearing infections. So you're not only having this very clinical production of mucus, and you're unable to really clear the infection, and you have all these symptoms. And so it really exacerbates the situation.
Yeah.
So that bias towards Th2, which we did discuss in the last episode when we were talking about Th2 versus regulatory immune responses. And so that was why that this vaccine apparently drove that Th2 response.
So we know a lot, and therefore the conclusion is we need an infectious vaccine. And there are a bunch in the development, but they're not ready yet. And I just... if you want to license a vaccine in a baby that's 6 weeks old, you have to do a clinical trial in that age. That's tough. So it's going to be hard. In the meantime, what we call passive vaccines have been developed, and that's when you give antibodies. And the first one was called Respigam, was marketed by MedImmune. And this is a polyclonal human antibody prep. And they just took serum from adults, and they enriched for antibodies that will neutralize RSV, because so many people are infected, everyone basically, but by a certain age, that you could just get serum and enrich for the right antibodies. That was licensed in 1996 by the FDA, and so that was used for a while. But then monoclonals were developed, and there are two of them. One is called Pavlizumab, or Synagis, or Synagis. Who the hell knows how to pronounce these fake names? Again, MedImmune antibodies.
Fake news.
They're IgG, and they're humanized. So, they're originally produced in mice, and then the genes are cloned out and humanized to make sure they're mostly human sequences so that you don't raise an antibody response against them. And those are directed against epitopes on the F glycoprotein. And we talked about monoclonals before, I think immune number 3. And so these apparently work. You can give these to kids who are recently infected and will modify the infection so that it's not as serious, but they're very expensive. And
they do it for high risk children, and they do it as a preventative too.
Yeah.
Because I actually had a preemie baby, and the first 5 months of life, they gave monthly injections of the Syncytial antibody to prevent the infection, because the idea was if the baby was born early, their lungs are not as mature at birth. And so exposure to RSV could even be worse than in a normal, healthy baby. And so they give this monoclonal antibody so it stays on board and will actually prevent infection during the first 5 to 6 months of life.
Interesting.
And the other thing, if with premature, you said 5 weeks, Cindy?
My baby was 8 weeks early.
8 weeks. And that really is the time when you see an increase in transplacental transfer of IgG, which maternal IgG antibodies to RSV. We can talk a little bit about that later, but they actually play a role in infants. And so if you have a preemie, those monoclonals may be even more important. But what I was also going to mention was if these monoclonal antibodies are more expensive, but 99% of critical infections happen in developing countries, I think that there's a mismatch there in terms of access to care.
Yep.
Absolutely.
Yeah. So
I think 13 years ago, I think it was $3,000 an injection.
I think part of the problem is one company is making all these, right? And so they kind of have a monopoly and they can charge what they will. The price has gone down. It used to be, I think, $25,000 for a treatment, and now it's much less, but it's still expensive, especially if you're going to get it monthly,
Right.
So people are working on making others. And the reason is because we know how to do this. We know how to isolate monoclonals. We know how to humanize them. So how can we improve on what we have? And so that's what this paper is trying to do. And they as you'll see, they look at antibodies in very young children as well as adults. But what is this somatic hypermutation? This is not something we covered before, so let's talk about that a little bit.
Sure.
When you think about all the microbes out there that infect us, right? Bacteria, viruses, fungi, anything. And we are pretty good at mounting adaptive responses, making antibodies and cytotoxic T cells. And that is amazing. If you think of it, just for the antibodies out there, you have to make a lot of antibodies. And it's estimated that humans can make 10 billion different antibodies. But there's no way we can have 10 billion antibody genes, of course.
We don't even have 10 billion genes in our genome.
That's right.
10 billion antibody genes.
We have a 3 billion base pair genome, and only 1.5% of it encodes for proteins. So What is going on? And this was one of the amazing mysteries of the immune system that I kind of saw unfold over the years as a grad student and a postdoc, and as soon as we were able to clone do recombinant DNA, it really, really accelerated. And I just was amazed at what happened out there. And the answer is that for antibodies, we start with just a limited number of genes, and then there are mechanisms to ensure diversification. And so this brings us to let's talk about the antibody molecule, the immunoglobulins, which we talked about before. They're made up of 2 heavy and 2 light chains, and each has a constant and variable regions. And these turn out to be encoded at 3 different loci on our chromosomes. So there's a heavy chain locus that's on chromosome 14. There are 2 constant genes and 44 variable region genes. And there are also other segments that are going to be put into the antibody molecule eventually. These are called diversity segments. There are 27 at this locus and 6 J or joining segments. And then the light chains are encoded on 2 chromosomes, 2 and 22. They also have 2 constant genes and many variable and joining segments, but no diversity segments. So we have genes for heavy and light chains in a couple of different places. And as B cells develop, and as during their development, these genes recombine to produce genes that can encode far more antibodies than are just present there in our DNA, and that's called VDJ recombination. And you can generate, in theory, about 3 times 10 to the 11th combinations of all the different segments. That's just amazing, right?
It's a lot. Absolutely fascinating. I think one of the coolest things about this is T cells and B cells are the only cells in the entire body that can do this. So T cells do it to their T cell receptor, and B cells do this to their B cell receptor or antibody. It's the same thing. And they take these genes and they just play mismatch with it. They just take chunks and just mix them together and match them together and make an encoding gene. And so the idea here that's really amazing is that the B cell receptor or the antibody gene is not actually transcribable and translatable in the genome until you do this.
Right.
Because it's in chunks, and you need to cut and paste the chunks together in order to make a functional gene. And it's that cutting and pasting together and choosing which of those, like you said, 44 variable genes or 27 diversity segments, it's which of those you choose and how accurately you put them and stick them together that allows you to have this amazing diversity. It's absolutely fascinating that this can happen.
I was lying awake at 3 a.m. and because my son is home from college and he makes noise and the dogs bark, all this stuff that normally, and I'm thinking I have to mention the RAG genes.
Yes.
Right? Recombination activated, what is the
activating
genes,
which was discovered, I think, the lady who did it came here a while ago. She was a Baltimore postdoc, I think, Marjorie Ottinger. Anyway, so this is one of the genes involved in doing this VDJ recombination.
Right. Mm-hmm.
And if you make RAG knockout mice, they don't do this. They don't make antibodies, right?
And they end up with no T cells and B cells.
Interesting.
Because if they can't make those genes come together into a functional form, the cell doesn't get a signal to stay alive.
Right.
Right. And if you're thinking about the location, trying to visualize where these processes happen. So the recombination events for B cells, they start really in the bone marrow, whereas T cells, we more talked about that happening in the thymus. And for B cells, you have these stages of B cell development that progress based on first the recombination of the heavy gene happens. And then after that, your pre-B cell is really an IgM isotype, which we'll talk about. And then you move to the light chain. And so when this happens, it's through this B cell development that's happening all throughout your life, essentially.
So when the cells come out of the bone marrow, Steph, what do they look like? Do they have antibody yet on the surface?
Well, they do. They have IgM and IgD. And then typically as that progresses, IgD is going to go away. And we have something called natural antibodies, which are IgM. And that plays into our paper a bit. But that would be a very non-specifically binding, but high. They have 10 different little fingertips or they have 5 arms with 2 fingertips on them that can bind up to 10 different sites. So they're special, but they don't bind as with high affinity as some of the others like IgG we talked about, but they are kind of the first antibody that naturally circulates.
So these B cells, when they come out of the bone marrow, do they go right to lymph nodes or do they circulate?
Well, they typically circulate, but they're going to go to lymph nodes where they're going to then interact with either dendritic cells directly or T cells to further maturate and do the.
Okay.
Yep.
All right. So that is that's how a lot of this antibody diversity arises, but it's not all.
That's right.
It's not all. There's more. And so, as Steph said, these B cells come out of the bone marrow with antibody on their surface, and in the lymph node they may encounter antigen. So a dendritic cell may bring in antigen, display antigen. And I think Cindy talked about that last time or the time before, right? And you called B cells stupid, right? And
poor B cells.
We better be a little nicer. It's a check and balance, right? Because a B cell doesn't know what it's making its antibody to, and so it has to gain more information and perspective about whether or not making that antibody and secreting that antibody is a good thing.
Right.
And the only way it's going to know that is if another cell tells it that. And it's often through a T cell. And so there's this idea of linked recognition where the antibody and that on the B cell is recognizing a native conformation of a protein that a T cell is also recognizing a part of that protein, but presented in a very different manner. But when you have the T cell activated and the B cell activated and they talk to each other, then the B cell knows that it should be making that antibody.
Got it. So when a B cell does meet antigen, brought in, say, by a dendritic cell, then the B cells start to proliferate. And they would only so if it's foreign antigen, right? If it's self, they wouldn't proliferate. Is that correct?
Well, the
B cells.
Right. They could, but that would be an aberrant response, and that would lead to autoimmunity. So
and that happens
ideally, it does. Ideally, it would go through a selection process after it comes out of the bone marrow and further selects for either, if it doesn't respond, they're either going to die. Yeah. Kind of like that Goldilocks and the Three Bears metaphor we used, where the intermediate binding is what's going to positively be selected. So
let's say it's a foreign antigen, the B cells start to proliferate. And at this point, the cells are dividing, right? And the heavy and light chain genes undergo a high rate of point mutation, and this is called somatic hypermutation. So it's just those genes, right? It's not every other gene in the B cell.
Only those genes.
Just those.
Only. And in the B cell, it's only the antibody genes. It doesn't touch T cell receptor genes.
Right.
And the T cells never do this. Right. So this is absolutely exclusively specific to these genes encoding antibody, and no other gene in the genome does this.
I don't know how you can call B cells stupid. This is so cool.
Okay, they're sophisticated.
She takes it back.
So you get one change per variable gene per cell division. And this is in part because of deaminases, right, that are mediating this process, which I think we'll talk about another time because they're pretty cool too.
Very cool.
And so you get even more antibody diversity. And you can imagine that if these changes lead to amino acid changes in the part of the antibody that combines with the antigen, you could get antibodies with higher affinity or with lower affinity.
That's correct.
You don't want the lower affinity ones, right?
You don't. And so there's this idea here where each time the B cell makes a mutation, it's going to go and test it. So it needs to hear information from the T cells to say, okay, yeah, that was a good thing to do, or no, that wasn't a good thing to do. Or they interact with follicular dendritic cells and try to bind their antigen. So there's various different interactions that can happen in the lymph node once the B cell is starting to do this process. And it's doing this in something called a germinal center. So people may have heard of a germinal center. And that's where all these processes that we're talking about today are happening. And so each time the B cell makes one of these mutations, it has to test, because the goal is to make these higher affinity antibodies. If it's the same, it'll get a signal and it may still live, but it may also try to make new mutations to see if it can do a better job. But if it actually made something with a lower affinity, it'll fail to obtain the same level of signal. And so it will probably just die away.
Yeah. Although it's interesting, I was reading recently, there's a lot of evidence mounting that these low affinity antibodies in the gut specifically are actually playing a role to keep the commensals in check.
Yes.
And that not binding with high affinity, because you don't want to attack your own commensals. And so it's interesting that if you for these pathogenic responses, high affinity is what you want to mature these B cells towards. But in other parts of your body where you want a tolerogenic effect, maybe the lower affinity is what's going to be positively selected in those environments. So I think it can be very context dependent on what organ you're in.
So is it safe to say, or correct to say, that if, let's say we're talking about lymph nodes in the gut, right? Would those have those kinds of antibodies as opposed to lymph nodes elsewhere, like respiratory tract, like where there's a microbiome also? I mean, it might have the same issues, right? No,
I think it's definitely because of the uniqueness of the gut, and that in the colon alone, I mean, you can have billions of cells, of bacterial cells. And I think that is very specific to the gut, but it's not to say that other organs, probably also thinking about like reproductive tract, would have this, but the gut, I think, is a pretty special place.
So this process is affinity maturation, right? So the somatic hypermutation, rounds of selection, as Cindy said, and that's affinity maturation. And for a virus, you want to have a high affinity antibody, so it can effectively block infection.
Right.
And that's what we're going to look at in this paper today. The other part which Steph mentioned before is that, when the antibodies first come out of the bone marrow, they have on their surfaces IgM and IgD, but you have to make other kinds of antibodies like IgG and IgA and IgE, depending on what they're needed for, and that is done also by recombination. You switch out the heavy chain region, and that way you can produce these different types of antibodies, and that switch is triggered by cytokines, right, produced by helper cells.
Mm-hmm.
And so now you can have, that's why, when you're first infected, you have IgM. And as Steph said, it's low affinity but higher avidity because you can have 5 molecules combining. And then during this affinity maturation, you're making higher affinity antibodies, and then you have them later, and then you switch to IgG, say, and that shows you that you have an older infection if you're doing diagnostics as opposed to IgM. That's
right.
Why if we find IgM against a virus infection, that means you're just been infected now.
That's right. And so this IgG or IgA or IgE, when you do that, we can call it class switch recombination or isotype class switch.
Right.
And the important thing to remember here is that the binding part of the molecule stays exactly the same.
Right.
Right. So you're still binding the exact same antigen with the exact same site.
Right.
But what you've done is taken that Y and chopped off the stalk and substituted a different stalk.
Yep.
And that stalk is what determines the functionality of the antibodies. So different antibodies have, as you said, IgM has 5 of them that come together. IgA can make a dimer. The other ones are more monomers. And each has a different, slightly specialized functionality. And so being able to make the same affinity and the same binding specificity, but swapping out the stalk regions allows you to diversify the functionality of that specific specificity antibody.
And just as a little plug for the last TWIP 148, we talked about IgE, which has the special property of being able to bind mast cells. And I just ignore IgE because I'm a virologist.
You should not do that.
Shouldn't do that, but I don't have any allergies and I'm a virologist. But for parasites, IgE is really important. And there's this cool story. This is awesome. For hookworm, so there are 2 kinds of B cells that make this IgE, B1 and B2 cells, right?
Right.
The B2 cell IgE will bind mast cells and cause it to, increase mucus production, and the parasite gets swept out, right? The B1 cell IgE delays it just enough so the parasite can mature. And then, it blocks mast cell activation by the B2 IgE. It's just amazing. You'll go listen to it.
That's amazing.
I was just stunned.
And so these B cell processes that we've been talking about all thus far are B2 B cells.
Yeah.
Right.
So those B1 B cells are sort of a special, unique cell type that makes antibodies in a little different way. It doesn't do as much maturation processes like we've been talking about.
So from what I got from that, they make polyspecific, and sort of the natural antibody that you have.
Correct.
Right. Early in life. And okay. All right. So that's what we need for this paper, right? Does that work?
Yeah. That's good.
All right. So cool paper, which is just illustrating the power of what you can do now. And please, I left out a lot of the technology, but if you feel like you want to talk about it, please do. All right. They took blood from 7 infants who had been hospitalized for RSV infection. 5 of them were less than 3 months old, and 2 of them were over 6 months old. All right. And they say 6 of these 7 probably had a primary infection. They take this blood and they analyze by flow cytometry the peripheral blood mononuclear cells. And they use I'll just say they use a reagent that can detect on these cells the viral F protein in pre and post fusion forms. So here's another thing we have to tell you. The F is a fusion protein. When the virus binds to the cell surface by the G protein, that triggers exposure of the fusion part of the F protein. So in its pre-fusion state, the fusion peptide is buried near the viral membrane, so it doesn't just react with any membrane. But when the virus binds to the right cell, which means it has this receptor that's engaged by G, then the fusion peptide shoots up to the membrane, sticks in the membrane, and catalyzes fusion. And there, it's the post-fusion state. So the two proteins look very different because one is, it's folded, the F protein is folded, and the fusion is down by the viral membrane, and the other, it's sticking up.
Yeah,
Yes.
when you have different conformations, if you can imagine, if you are thinking about that process and you use your hand and you're moving your hand, you can see that different parts of your hand are going to be exposed. And so if antibodies are looking for epitopes on those different conformations, it's going to change depending if it's pre-fusion or post-fusion.
Right.
Yeah, the antibodies are really only able to interact with those things on the surface of whatever it is, right? So if something is buried on the inside, that's a motif that a T cell could see, but it's not something that a B cell could see.
So they're basically looking for B cells that are specific for F in its pre or post fusion states. That's what they did in this experiment. And they can have numbers, and they can say that the frequency of class switched B cells that are specific for F is lower in infants less than 3 months compared with greater than 6 months. The numbers look great. You can look at them and you can really see this. And that's something we expected, right? This is nothing new, but you're showing it here for this particular viral protein. So then they single cell sort between 1 and 300 of these F reactive cells from each donor, and they amplify out the variable heavy and light sequences by PCR and sequence them. And they want to look at now somatic hypermutation, because they can look at that and see it by sequencing of all these different kids, young infants versus older infants. And you can say how much somatic hypermutation, and they can see that it increases with age. It's another just beautiful thing that you couldn't do this 15 years ago, right?
Right. And this might not have been predicted, right? Because what we knew about it was the somatic hypermutation increases during the infection.
Mm-hmm.
Right? So once you have exposure, everything we talked about in the introduction is once you have the exposure, the B cells start to do this as they proliferate, and then they test an affinity mature and they bring out the highest affinity ones. But what this is saying is that the young infants don't actually do that.
Right. And it could
as much.
As much as the adults. And it could be because in infants, there are a lot of different co-receptors on the surface of cells that are also needed for really optimum somatic hypermutation, and they are lower in infants. And so this could be a reason why you don't see this in infants as much. And it's going to kind of bias the response differently than even older infants, older than 3 months.
So they do see some mutations in these variable regions of the youngest kids, but they say the frequency is far lower than the ones 6 months and greater. So just in this 3 month period, the ability to carry out more extensive somatic hypermutation has increased, which I think is amazing, right? It's like 3 months. It's really big.
Yeah.
All right. So then they also, this kind of sequencing can show you there are different kinds of variable genes, and often there's a bias in which ones are used in the response to certain viruses, and they see that here. They see a bias. And unfortunately, for me at least, the way they name these V genes is very tough. And I look at them and my eyes glaze over. I can't remember because they have nothing that's impressive. But I'll just say that the V gene usage is biased, and we'll come back to that. Again, the F-specific antibodies, remember, they have to use different variable region genes, but they don't use all of them. They only use certain ones. And it kind of makes sense if you think about it, because these have to combine with a specific viral sequence, and you can't just use any variable gene, right? It has to be
right. There's going to be preference.
Yeah. Okay, so next they look at the binding affinity of these antibodies. And here, the cool part is they actually put these in yeast. They put the genes encoding the antibodies in yeast, and they produce the proteins. So you can't complain that I only have 200 microliters of blood from each baby, because it doesn't matter. You don't need to use it. You've cloned out the antibody genes. So they make IgG antibodies in yeast, look at binding affinity with virus, of course. The youngest infants, it's pretty good. The cutoff for low and high affinity is 5 nanomolars. So less than 5 nanomolar is pretty good binding affinity. And 24 to 33% of the antibodies of the young infants had high affinity. They're mostly against pre-F. And the older infants, that's again 6 months and older, they had 45 to 90% of high affinity antibodies. And these were a mix of pre and post-F. And we're going to...
Now, would we think that antibodies against pre-F or post-F would be better or worse?
You would want pre-F, right? Because that's going to be exposed before the virus is hitting the cell.
Mm-hmm.
But post-F, we'll see later, post-F antigens cannot induce pre-F antibodies.
Mm-hmm,
so that's problematic. So you would like to have pre-F. I believe post-F will also neutralize as well. It will prevent subsequent steps in entry. But I think you would like to have pre-F. Let me look ahead here and see. These monoclonals that are made, I did write here. I don't remember if they're pre or post. I think they are pre-F because they're targeting one of the antigenic sites on pre-F.
Well, and we learned that, I think we're going to talk about it more, but we learned that with the vaccine, it was mostly targeting post-F.
That's right. That's
antibodies,
right.
and they're non-neutralizing. Right. So I'm assuming just
you're absolutely right. That's it.
Right.
Yep.
Right.
Yep. All right. So then that's binding affinity, how tightly they bind. But just because an antibody binds tightly to a virus doesn't mean it's going to neutralize it. And by neutralize, we mean blocking infection.
Right.
So it's a term that we throw around, but for most people, it doesn't make any sense when you say an antibody is neutralizing. And I learned that a while ago. I was teaching in the class, what does neutralizing mean? I said, oh, sorry. And from then on, I learned you have to define everything. Neutralizing antibodies. So about anywhere from 11 to 50% of the antibodies from each infant had neutralizing activity. Some of them is really high, which means you can dilute the antibody way out and still get neutralization. That's what we mean by highly potent activity. 20% of these neutralizing antibodies had no V gene mutations. In other words, they didn't undergo somatic hypermutation. Okay, so you can get neutralization with just the germline sequences, right? No mutations.
Right.
Yeah.
Which is very cool. Right?
Yes.
Now, if you have somatic hypermutation, you get more higher neutralization. So the fraction of higher neutralizers is higher in antibodies with SHM than without.
Right.
But you can still get neutralization. And most of the highly neutralizing antibody bound pre-F, but not post-F. So pre-F is good for highly neutralizing. And nearly all the neutralizing antibodies in the very young infants are pre-F specific. That's less than 3 months of age. So pre-F seems to be the antigen of choice, to answer your question now.
Mm-hmm.
All right, next, what epitope? So an epitope is the part of the protein. Cindy, how big is an epitope roughly?
Oh,
for a protein?
It can vary because it's a three-dimensional structure on the surface of whatever thing the antibody is recognizing. So what's interesting about that is it can be contiguous. So you can have 10, 12 amino acids in a row that are recognized.
Right.
Or you can have them folded and wrapped around and recognizing those same, however many antibody amino acids they are, but they're not contiguous, but they're close together in the three-dimensional surface. So exactly how big that is, I don't know size-wise. Do you know, Steph? I'm not really sure. I think it varies.
So, I mean, the B cells, it's going to be larger. So the difference then between T and B cells is T cells need it to be linear. They need to be shorter. B cells that can have that conformational structure. I'm thinking B cells, like 30 is amino acids, and then T cells is less. Is the number that's popping in my head, but
9 to 11, 13-ish. And
so I just want to give people the sense that this is a small part of a big protein.
It is a very small part of a much larger protein. Yes.
So if you imagine this F protein, they're just parts all over it that antibodies could bind to. And those are the epitopes that we're talking about. And something people have done for years since monoclonal antibodies were invented, and you had the ability to do this, is to say, what are the epitopes recognized by antibodies that either just bind or bind and neutralize infectivity? It's really important.
And I guess for a while, what they did was competition, right? So if you had two different antibodies, you asked, could they both bind at the same time, or did they compete for binding? And if they competed for binding, you knew they bound something that was overlapping or very close to each other. Right. Whereas now, I guess it's even easier because they just crystallize everything, right?
Well, they did that eventually here, but before they did that, they actually did competition.
Yeah,
both is good.
They had a panel of known antibodies specific for particular epitopes on the F protein, and they simply did competition studies with the antibodies that they had made from the babies. And so they found that, so there are, and again, I was thinking about this at 3 a.m. because they have a funny way of labeling. You just can't sleep, so you think about,
are
you supposed
immunology,
to, they say if you want to fall back asleep, you have to think calm thoughts, and you can't be worried about what you have to do, So I try and think about science, which is always calming.
Yeah.
Antigenic sites on glycoproteins, they have 0, 1, 2, 3, 4, and 5, kind of Roman numerals, but the 0 is not a Roman numeral. So I was lying awake thinking, why do they have? It's a 0 with a slash through it.
No. Yeah.
So who knows? But anyway, there are 6 antigenic sites that have been mapped. They have the 3-dimensional structure of the F protein, and you can see exactly where they are. And so they can map these new antibody specificities to these antigenic sites. And what they find is that the youngest infants mostly make antibodies against site 3, and these are not exclusively, but mostly, and these are mostly pre-F, and they are of medium to high neutralization activity. The older infants made more against site 1. These are post-F site 1s, and these are weakly or non-neutralizing, or they made them against site 4. And that's a pattern that is seen in adults, which also make more antibodies against sites 0 and 5, which are also low in infants. So you see distinct patterns of antibodies made in the very young infants and the older infants and adults, which is interesting. Because again, if you want to make a vaccine that induces the right, quote unquote, right antibodies, right, that are neutralizing and highly neutralizing, you need to know this. And you especially need to know it in babies, which are the ones that you really care about.
Yeah, there was one thing that I was thinking about in this whole paper, and that is that they started with these infants that were hospitalized.
Right.
So they're the ones that are not controlling this infection well.
Yeah.
And how does that influence the bias of what types of antibodies that they're reading out? So if they were able to, which is not going to happen because you don't have the sample population there, but if they took young infants that didn't have such severe disease and they characterized the antibodies, do we think it would be the same or different? Might it be more like the older infants or the adults, or it's not clear to me exactly what would happen?
Yeah, that's a good question. I assume it would be similar to adults, right? But they're even different from the older infants. So
right.
I don't think we know. I mean, they did look at just antibodies from cord blood of kids who didn't have infection, right? We're going to see that later. But that doesn't answer your question. So it's a good question.
My question can wait till the end because it's a similar question, but I'll wait till we've gotten more through it.
Okay, next we're going to look at these antibodies that aren't hypermutated. They lack somatic hypermutation. What were they recognizing? Because remember, the younger babies, which are particularly at risk, they don't do much somatic hypermutation. So you want to know more about those antibodies. Mm-hmm. 27 out of 33 were against site 3. And half of these were from naive B cells and half were from memory B cells. So these are cells that are already there, right?
Right. So they had talked about that these are going to be most likely primary infections in these infants, but there are memory B cells there. So, you're thinking about, would it really be a primary infection if there are memory B cells there?
So are there no memory B cells unless you have an infection and a response?
I mean, there can be so this is my question. Were these infants breastfed? Because there are memory B cells in breast milk to RSV, and actually one of the vaccine strategies they're looking at is maternal vaccination, which can bias the antibody repertoire. So my question would be, were these infants breastfed? Did they have maternal antibodies? So the memory B cells could come from breast milk, or this may be later in infection where they have already developed some memory B cells. I think that's the problem with not really knowing what stage of infection they're at.
Yeah.
So there's also this idea that there's these innate memory T cells, innate T cells that have this memory-like phenotype. And so I wonder also if there could be some of these B cells that sort of have an innate memory-like activity and surface marker repertoire that may be designed for this exact type of thing where during the early developmental stages where you don't have the fully formed immune system, could there be these B cells that are good at making lots of antibody early and sort of have this memory-like phenotype? I don't know because I don't follow the literature in depth, but it could be that may also be that type of cell as well. I don't know how well that's been characterized.
I think that blurs the line between we often, when we teach and learn immunology, it's innate and then adaptive. And I think what we're more appreciating now is there's a lot of cell types that are bridging that gap. Even just discussing these kind of innate like IgM antibodies, B cells that produce those antibodies. So it could be, Cindy. I think that could be true.
And just to remind you, the site 3 was the preferred site in the youngest infants, and you get neutralization, medium to high neutralization activity with antibodies directed against that site. Okay, so next, do we see these antibodies in naive B cells? So to do this, they cloned out the F-specific antibodies from cord blood and from healthy adults. So the cord blood, I guess you get from, I'm not sure where they got it from. It's not from these babies, I believe. It's just from
I would doubt it, yeah.
General population, right? Because they wouldn't have known that these cord blood is taken, I guess, at birth, right? When they're doing studies. So,
yes.
So this is some cord blood that was from some other study in healthy adults. And they find some high affinity antibodies in both sets against site 3. So no somatic hypermutation. These are just naive B cells. And you see highly neutralizing anti-F antibodies in the germline, which is pretty cool. And
it is.
We'll get back to that. They do some crystal structures, as Cindy said. They just, you want to map epitopes, just do the crystal structure of the antibody bound to the protein. It's amazing that you can just crank these out. It used to take years to do this. So they do one of an antibody targeting site 3 on the pre-F, and there are several analyses of these. They do a couple of them. The bottom line is this shows you why there's a bias for certain variable genes, because in most of the cases there are tyrosine residues in the antibody that contact the pre-F, the viral glycoprotein, and they say this looks important, so they change the tyrosine to an alanine, and that reduces the binding affinity of the antibody, right? And so you would need to have V genes that have tyrosines at these positions, and not every V gene would. So that explains the bias.
Right.
And they go through that for a couple of different antibodies. It seems to be a tyrosine often that is involved with this.
Because I think that one of the reasons why that is because it's a bulky group and a hydrophobic group. So when you think about a lock and key coming together, the more it fits into a pocket, the stronger that's going to be.
Right. So that's basically what I wanted to tell you. In the discussion, they talk about this idea that humans may have these what they call functional germline antibodies. There's no somatic hypermutation, but they can strongly neutralize, in this case, RSV. Maybe because this is a pathogen you're likely to encounter early in life. They found these antibodies in all 7 infants. They found them in cord blood cells, adult B cells, memory cells. So, maybe I don't know how that would work, because this virus would have to be around a long time in order to select for these kinds of immune genes, right?
Right. And that's where I think we need TWEVO, a little crossover, because I was thinking about this, and I think TWEVO right now is one of my favorite podcasts because I'm just not evolutionarily trained to think often that way, but it would have to be around for a long time.
Right.
And when we look at, I think, Vincent, you're going to bring it up, but HIV, they don't have germline antibodies that react with HIV.
That's right.
And HIV has not been in people for that long. So I don't know if it has to do with the fact that we are evolving with these pathogens, and that's because why else would it be in the?
Oh, it'd have to be. It would totally have to be that somehow this virus RS imposed a selective pressure. Right. Mm-hmm. It killed off kids who didn't have germline antibodies that could recognize it. But then those genes have to propagate through the human population. Yeah. And that takes forever. It takes thousands and thousands of years. And it probably happened a long time ago when there weren't that many humans on, Homo sapiens on the planet, because it would propagate through that population a lot faster. Because someone told me once that if a gene arose today that was some advantage for humans, it would take 10,000 years for it to spread so that you would actually detect it in the human population. So, the HIV, it probably arose in the 1900s, right? And we haven't had any selection at all. No. But the RS must have been around pre-Homo sapiens in order for this to happen. It's an interesting idea. The other thing that comes out of this is that you want any vaccine, if you're going to have a non-infectious vaccine, you would have to use a pre-F confirmation, right?
Right.
Because post-F makes non-neutralizing antibodies. And as Steph said, the failed 1960s vaccine induced mainly post-F, probably because of the treatment. It just flipped it into a post-F confirmation, and that induced non-neutralizing antibodies.
Right. I remember seeing something in the paper about if they make the recombinant, I think it's if they make the recombinant protein or if they just purify the virus, the pre-F has a tendency to unfold before it should, and then adopt some post-F-like conformation that then doesn't really work well for vaccine development. So I guess one of the ideas would be like one way people would make a vaccine from this type of information to say, okay, we want to make a vaccine to the pre-F, and here are the sites that we've identified that are really good for neutralization. So let's take a chunk of that protein, or let's make this recombinant protein, and then let's inject that into people with some adjuvant, and they'll make the right antibodies.
Right.
And I think the problem is that if you make that protein and it folds incorrectly, then you're going to get antibodies to things that you don't want or may actually be detrimental.
Well, even in vaccinating infants, I mean, yes, the antigenic binding sites and whether we're trying to get pre or post F, of course, is a consideration, but there are a lot of other aspects of infant immunity that are just, they either, and it's, infant immunity has been thought of as for a long time that they just don't have any of the cells and cytokines and responses that adults have, but actually they do have, for instance, they can respond, their T cells can respond to certain antigens at the same capacity, but they have maybe this immune regulatory blanket over them, and it's thought to be because they need to be colonized by the incoming microbes. They need to be able to kind of set that colonization of the gut and other parts of the body. So I think it's a little simplistic to think that just targeting pre-F would be a good vaccine candidate, maybe including a TLR agonist, because we mentioned before toll-like receptors were degraded in the vaccine, and maybe that was why they weren't stimulated in the correct way. So infant vaccinations is very difficult, although maybe vaccinating mothers with a pre-F driven vaccine could then transplacentally transfer those antibodies so they're protected. But I looked it up, in RSV antibodies, their half-life is only about 1.5 months. So maybe having to increase the antibody half-life would be another. So there's a lot of different things that go into vaccine development, but I think this paper does a good job of showing us at least one aspect how we could improve.
And it got us to talk about somatic hypermutation.
Yeah, absolutely.
There's one thing that we haven't touched on yet that might be just worth mentioning is that we're talking about placental transfer, and also in the last paper we discussed in the previous immune episode, we were talking about transfer of antibodies through milk, and sometimes that's a great thing, and a lot of times that's a great thing because if the mother is exposed to something, she's going to transfer antibodies that will then be protective in the infant. But the downside to that is that if the mom's antibodies work really, really well, the infant may not develop the immune response the same. So it's this idea of maternal antibody interference.
Mm-hmm.
Yeah, and it definitely, I think RSV, measles, we talked about a little bit last time, but there's some mechanisms by which the antibodies block the epitopes of the vaccine antigen, so the cells can't even see it, and amongst other things. So, yeah, it's difficult. The other question I had was, in terms of the neutralizing capacity of what Vincent was talking about, the natural IgM antibodies, what would be, how could you increase natural antibodies? If in the baby, those might be more protective and maybe less detrimental. Is there a way to do that? Because you can't vaccinate, because then you're going to have somatic hypermutation if you do that.
I think they mentioned that in their, they say maybe expanding these germline cells is a goal, but I don't know how they would do that.
But how, right? Yeah. They don't really mention how.
No. I mean, they say infant vaccination, but I don't know how you would do that.
Yeah.
But I mean, really the thing that kind of bothers me in the end is that it must be really hard to recruit babies for vaccine trials, right?
Yeah.
Because, if you're an adult, you say, yeah, I'll do that. You can give me this vaccine. But your baby, most people do not want to have anything done to their baby. So I think it's really hard. I mean, you're going to find people, but it's not like you can just do them left and right, I think. So that's another consideration. Anyway, I thought that was a fun paper.
It was.
Good. I've been thinking about RS since I was a grad student because of this vaccine, and this is really cool. And the technology is amazing. There are a lot of things we glossed over, but
totally
the
single cell PCR to PCR up all of those different gene segments and look at the hypermutation and everything.
Amazing.
It's pretty amazing.
Yeah,
it's really neat.
We could even dedicate an episode to. We could pick out a couple things, maybe ones you mentioned, and we could explain them sometime.
Totally. We have to do tetramer staining, right? Because that's that's going to pop up in so many papers and other things as well, for sure. I bet people would like that. All right, let's do a couple of emails. Okay. Cindy, can you take that first one from Bowden?
Bowden. Okay, I was going to ask how you would pronounce that. Bowden writes, Dear Immuners, it was a very balmy day today in Chapel Hill in Durham, circa 64 degrees Fahrenheit. Boo. It's not warm here. So I decided to send you a letter to point that out to you and make you jealous. You did. I welcome your new podcast. Immunology is indeed an abstruse subject, especially for physicians. You began with monocytes and macrophages, the most interesting and diverse cells in the human body. I agree. With reference to light and lymphocyte immunology, multiple sclerosis varies consistently with latitude. Its prevalence increases with latitude. This is largely true for the northern hemisphere and less for the southern hemisphere, since the northern hemisphere has a much broader latitude distribution and a much larger population. The information in the southern hemisphere is much more scant, except for Australia and New Zealand. The reasons for this are not fully clear, but probably relate in part to UV light exposure and HLA-DRB1 induced susceptibility. An additional and corrective factor is most probably intake and level of vitamin D. And so before reading the rest of the letter, I thought it's interesting to interject here that vitamin D is actually really important in regulating the immune response. It's critical for those regulatory T cells that we've mentioned here and there, and for balancing the type of immune response. And vitamin D can suppress certain types of immune responses that are associated with autoimmune diseases like multiple sclerosis. And this includes a cell type that we call Th17 because it produces a lot of the cytokine IL-17. So this idea of vitamin D and autoimmune disease and these different types of T cells makes a lot of sense. One of the cool things about vitamin D is how humans get vitamin D is by UV light exposure. So when we go out in the sunlight, we make vitamin D in our skin, and then it helps to regulate our immune responses. And so there's also obviously an impact on your ability to produce high levels of vitamin D based on your geographic location, because the more of a northern latitude you are in the northern hemisphere, the less light exposure you have. And I guess if you're in a colder environment like we are in Ithaca, you wear more clothes covering you up, and so you have less light exposure. So all of these things are connected. So back to what the the email says, it says, I thought this might provide a clinical correlate to your Immune 2 podcast on immunology and circadian rhythms with reference to Immune 1. So he's linking lots of our shows together. Bats may be resistant to Ebola because of their high metabolism during flight. And this gets into thermogenesis because the rise in temperature produces changes in metabolism and critical proteins providing resistance to viruses and other mammals are susceptible to. This probably needs more investigation. And I would agree with that. So there's a link that we can put in the show notes, flight as fever. So metabolic increases in temperature change the ability of the bats to make different cytokines, and also just their temperature, which we talked about, regulates functionality of immune cells and their trafficking in and out of lymph nodes, for example. And also the idea of circadian rhythms connects back again to your light exposure, because if you're in a more northern, like say Sweden or Finland or Alaska, and you're in the dark for 24 hours a day almost, certain times of the year, that's going to affect your ability to make vitamin D. And so all of these really fascinating things are interconnected here. And so I think it was really great that Bowden mentioned this, and we could connect all of those things together thinking about this. And so he ends with, thank you for your excellent work and Godspeed in your project.
Thank you. Thank you, Bowden. Check out TWIV, the latest TWIV 484.
Yeah.
We did a paper where they find that a critical component of the DNA sensor called STING has a point mutation in a critical residue in bats that dampens the response to virus infection.
Yeah, it changes their production of interferon, right?
Yeah, it's very cool. All right, I will take the next one from Lauren.
Are you ready?
who writes a very long email about histoplasmosis, which I won't read, but it will be in the show notes in the email section. You can take a look at it. But Lauren is writing about her and her coworkers had an outbreak of histoplasmosis, and they're all, she says, immunocompetent. They got disseminated histoplasmosis. She thinks she got it from roosting bats, and then goes on to talk about the pathogen, the outbreaks, and how it's often ignored and misunderstood by the medical community.
Now, histoplasmosis is
it's a fungal infection.
Disease from a fungus.
So it's a fungus, very common in the Mississippi and Ohio River valleys.
Yep, but I guess I did not grow up in the River Valley, but I visit there quite often, and
I'll bet you're infected. You're infected.
I bet I am. But does it come from bats though?
No, well, it's in the soil, so you can just get it from the soil. 90% of the people in that area are infected. Also, other parts of the world too. It grows very well in soil enhanced with bird or bat droppings, so there's a lot of nitrogen. And clusters of infection occur in people who visit caves, or if you're near an old building that is demolished. For example, biggest outbreak ever, 100,000 people got infected in Indianapolis who were near an amusement park that was being demolished. So, it's full of this fungus. You inhale the spores, it transforms into the yeast phase inside of you. So it grows in nature as a mycelial form, the nice filaments. You inhale spores, it transforms to a yeast. Most people get rid of it, but the cells can remain in you for many years. And if you're ever immunosuppressed, you can have a reactivation of this infection. So an immunocompetent person will control the infection, not get disease, but have reactivations later on. But of course, if you're immunocompromised, if you get AIDS or some immunosuppressive drug, you will have reactivation. You can get serious disease, and you can get very serious stuff happening, and particularly in older people. So this is well known, and Lauren goes on to talk about how it's difficult to get treated for this in immunocompetent people. And she's written a book, so that's the last thing I want to see. She's written a book about her experience called Batshit Crazy.
I love the title.
Because bats shed the fungus in their feces, and it causes delusions and hallucinations. So the fungus can get in your brain if it's not controlled, and yes, that can happen. Yeah. So it's unfortunate that you've had this experience, but there are certainly plenty of doctors who can treat this. Steph, can you take the next one?
Yeah. Anthony writes, my impression is that for most of your audience, possibly including your colleagues, understanding immunology requires something of a cheat sheet. My suggestion is not to do this in the body of the podcast. For each show, include a second recording of a few minutes of up to speed covering definitions of terminology and an outline of concepts that will appear in the episode. That way, people like me can listen to that first, and those well-versed in immunology can skip it. For an idea of another show, perhaps there can be one on new drugs, drug discovery, and other pharma topics. I think that in addition to your audience of biological professionals and the interested public, a drug podcast could be a resource for industry and investors, potentially many more people than the ones that would interest advertisers. If memory serves me correctly, while at Microsoft, Bill Gates' hobby was buying biotech stocks. The story goes that he holed up in a hotel room one weekend to study molecular biology and so be able to intelligently invest. Few who invest in pharma might be able to do that, but they can listen to a podcast while commuting. So I think our goal is to, in the beginning, introduce an in brief of what we're going to be talking about, giving you terminology and definition. Then if you feel like you are seasoned enough in immunology, you can skip forward until we say, okay, here's the paper, but you can, of course, listen to it. We thought about doing, different immunology 101s, but it just seemed to be, we couldn't keep you up to date on what we're going to be talking about that way. So best to do it as an intro to the podcast. That was, I think, our philosophy.
Yeah, we'll see how it works. See if people like it. You could skip through if you don't. Yeah, as you say, you don't need it. Cindy, you can take the next one.
Sure. So Kevin writes, howdy immune gang. Kevin from Audio Immunity here. Quick pronunciation guide. It's audio immunity. I think I said it wrong. So audio immunity, same number of syllables as autoimmunity. I wanted to respond to Steve's question from episode 3 about our podcast. Yes, our episodes are in a bit of a random order, but I've tried to be diligent about adding at least a couple of tags to each post that include the main topics discussed. Things like MHC, B cells, etc. There's even a tag cloud along the right side of the immunity.org/audioimmunity that lists them, and Steve can click those links and filter just the episodes that have those tags. I totally agree that having something more systematic would be useful, and any listeners should feel free to send us an email to let us know if some episodes have tags missing. But that might go at least part way to helping listeners find episodes that cover each topic. Incidentally, our most recent episode covers some similar ground as your episode 3. We were talking about efforts at generating a vaccine for rhinovirus. Loving the show so far. Keep up the good work. So this is someone that you had contact with, right, Vincent? This audio immunity people.
Yeah, and we mentioned them, and Steve wrote in a letter requesting some order, right? But I don't think who knows where to start, right?
We're going to be very organized. We won't have that problem, right?
Oh, right.
Well, I think you just dive in, right? And like today, you start explaining somatic hypermutation. And if you don't know what a B cell is, you can go back a little further. And we must have explained it somewhere. But I think as he said, tags are going to be okay. But we're not going to go in any particular order. We're going to find papers that interest us and then give you the background that you need for that.
And I'll tell you something that even when I teach immunology in a somewhat coherent fashion, it's such an interconnected web that it's impossible to teach it in a linear fashion.
Yeah, that's right.
Students always ask me, why can't we just cover it in a linear fashion? I said, because it simply doesn't exist that way. And so if you introduce topics in one order, you still have to circle back and then explain them in more detail. Because if we started with T cells and we tried to explain everything you ever needed to know about a T cell, you'd get completely lost because it doesn't exist in a vacuum. It exists in context with all of the other cells, and you need to know those cells so that you know how they talk to each other and why that's important.
Yep. The next one's from Richard. Hi, esteemed professors. Hey, Steph, you got a promotion.
I know. I saw that. I was like, well, I won't say anything if he doesn't.
It's okay. You can be a professor. It's fine with us.
Someday.
Firstly, the weather here is 7 degrees Celsius, dry and sunny with 7 kilometers per hour wind. You ask about subjects, listeners would like to hear about, I would like a 101 episode on antibodies, the function of these in the wider immune response. I understand they are Y-shaped and the basics, but how do they get formed from presented antigens, etc.? Well, I guess we covered a little bit about that today.
Yeah, this episode was a good one.
Although it could use a little more, but we'll get to it.
Sure.
I'm a water treatment engineer and longtime listener to TWIV, TWIM, and now Immune. So not a scientist, though I'd like to think I have a fair grasp of the subject by now. For example, I've looked through all your virology courses on iTunes U, as well as trying to listen to audiobooks on science whenever possible. Thanks for your time in producing all the podcasts that you do.
Yep. Jolene writes, greetings, Immune hosts. I love your podcast. Can't wait for the next episode. It has been exactly 1 month since the last one was released. This may have been an earlier email. In the first couple episodes, there was a discussion of how temperature influences immune cell activity. I was shocked and delighted to hear about this on my early morning dog walk. So many incredible things happening inside our bodies all the time. Here's where my friends would ask me if this is why people can catch a cold, the virus, by going out uncovered in the winter. I know that there are other factors that play into seasonal illness. Do we know much about short-term versus long-term cold air exposure affecting the human immune system enough that we are more likely to become ill? I'm especially wondering about extremities and the immune cells in your respiratory tract. Must also echo the desire for some Immunology 101. While this could be a great resource for college students, I especially think it could be invaluable for the regular people out there. Please count some extra votes for recording those episodes on things like complement, antibody isotypes, differences between phagocytic immune cells, subpopulations of immune cells, and soluble immune factors at different body sites, T-cell maturation for self versus non-self recognition, etc. Okay, maybe some of those topics are beyond 101, but I would happily listen to them on the podcast. Best. So in reference to the first couple questions she had, I do not think that when we were the first episode that we discussed, when we saw a paper described the circadian clock rhythms of lymphocytes entering into the lymph node more during nighttime in mice, of course, and then in the blood more during the day. That can impact things like lymphocyte migration. But, she's mentioning, the one you did, Cynthia. That's the first one, correct? Yep. So I don't know if you have any insight, but I don't know if in terms of just being outside for some time in the cold could affect that.
So there is actually a very cool study by Akiko Iwasaki at Yale, where they looked at how the common cold virus replicates at core body temperature or at slightly lower body temperature. So one of the things we don't think about with our own body temperature is, at least for me, for sure, our hands are much colder than our core. And often our nose is colder than the bottom part of our lungs. And what they found was that the virus actually replicates better at slightly lower temperature. And so the immune responses were slightly better when you're at core body temperature. And so the idea here was breathing or having the colder temperature in the nose allowed the virus to gain a foothold. And so even though, like I've been saying, and I can't remember exactly when her study came out, but I was saying for a long time, this is a myth. There's always this, I don't know what you call it, where your mom says, don't, you have to wear a coat outside. If you get cold, you're going to catch cold. And
right.
I've been saying, it's a myth, which is good because my kids play in the snow in shorts. But the reality is when they look at it in a more nuanced way, it does seem that there is a temperature dependent replication thing that might be associated with during colder weather when you're breathing colder infected air. It might promote slightly the ability of the virus to gain a foothold.
Yeah, I was looking it up. That were in 2015, and it was Ellen Foxman was Akiko's postdoc, who I'm pretty sure has her own, is now an assistant professor at Yale, starting her own lab. So yeah, that's, if you search really Foxman, Akiko, you can find it, 2015. Yeah, there it is. Mm-hmm.
All right, let's go to some pics of this episode. Steph, do you have something for us?
Yeah, so I posted in there, and basically it's called the REACTOME project. I've known about this for some time, but I was brought back to it because they have a lot of really neat free icons and free little immune cells or proteins, or you can go on their website and look up all their different icons that you can freely use for your presentations. And I just like that there's access to really nice looking science icons instead of trying to spend
look at the antibody.
Yeah, check it out.
Cool.
Taking a lot of time to make your own can waste a lot of time. So they have some available. And the general website is really neat because it's this open source, open access pathway database where you can go in and visualize different bioinformatics in terms of different systems biology. So you can click on an organ or a pathway and see visually what happens in that pathway. And then you can actually access that data. So it's kind of like joining together all of this big data we get from bioinformatics and signaling pathways in the cell and be able to access that. So I thought it was pretty neat. You can play around on there and find different things.
Oh, the icons themselves are just great.
Yeah, they're neat.
I mean, there are a lot of just buttony type things, but there's some molecules drawn out, which you always need these. This is great.
Right. Did you find poliovirus? I'm looking now.
I didn't look for it, but there are lots of protein molecules, which are good.
Yep.
And, they're icons. They're not supposed to be all that accurate, but they're good enough for a lot of uses.
Yeah.
That's great.
Mm-hmm.
Very good tool. Thank you.
Good. Sure.
Cindy, what do you have?
Yeah, I have an article that was in the Atlantic by science writer Ed Young. And the title of his article was, I spent 2 years trying to fix the gender imbalance in my stories. And the idea here was this astute individual realized that there's a bias in who science writers interview for quotes. And so he chronicles his science writing experience in the realization in his own imbalance in gender and what he did to correct it. And the spoiler alert is that it wasn't easy. He said it was more time, but it wasn't so onerous to ignore the imbalance. And so the idea was if you contact people for quotes, men will tend to respond more frequently than women. And so we're underrepresenting very talented, intelligent women who have important things to say. And so I just thought that it would be interesting to highlight one thing, a quote from the article. It says, she is right. Women in science face a gauntlet of well-documented systemic biases. They face long-standing stereotypes about their intelligence and scientific acumen. They need better college grades to get the same prestige as equally skilled men. They receive less mentoring. They're rated as less competent and less employable than equally qualified men. They are less likely to be invited to give talks. They earn less than their male peers, and they have to deal with significant levels of harassment and abuse. And we've heard that a lot in the media lately. Gender biases are also entrenched in the media where, in the words of the sociologist Gay Chutchman, women are being symbolically annihilated. This seems really horrible, but the idea here is that science communication is so important, and we think about it in gender from this article. You know, if women are less likely to respond when a journalist contacts them for a quote about a recent science article or whatever, they're missing that opportunity to speak. But I think it's bigger than that, because it's scientists in general. If we're not willing to speak up, somebody else will speak up for us, and we're going to lose our voice. Totally. And so it's true at the individual researcher level and more broadly in science. I think if scientists are not the ones talking about scientists in the media, non-scientists are going to be talking about science in the media.
Right.
And so there's going to be a significant bias. And that's where we get into non-fact-based propagation of information.
Completely agree on all points.
Absolutely. And I think especially in terms of looking at different stereotypes based on either gender or race, sexual orientation, I see a lot of pushback even in the scientific community. And I think that a lot of the rebuttal would be, no, we should not focus on that. It should only be based on ability to produce. But what that fails to consider, of course, is the well-documented we don't live in a vacuum, right? I mean, there are biases and stereotypes over history that play into this. So it's not as easy as just saying, oh, you just pick the best science. I mean, there are forces that we have to, and our own biases, I have to work on it all the time. It's something that I think as scientists, it's quantifiable and we should be aware.
Totally. All right. I have an article in the New York Times from February 9th by Don McNeil. He's a health writer for the Times. It's called Trump, Tell Us About Your Flu Shot. And the whole idea here is that we need to have public health leadership in this country to save people's lives. And McNeil says, if Trump can claim credit for the fact that no Americans died in plane crashes in his first year in office, he must also shoulder some blame for the high flu hospitalization rates that were and are predicted for this year. So it's an interesting article. I highly recommend it. The part that I want to focus on is, has he had a flu shot? In a 2015 radio interview, he boasted that he had never had one, saying, "I don't like the idea of injecting bad stuff into your body." Nevertheless, he appears to have had one this season. He was examined by a White House doctor in January who said his vaccinations, including seasonal influenza, are all up to date. So he says, I don't like the idea of injecting bad stuff to satisfy his supporters, of course.
Right.
Yeah, and I think he has, during the campaign, also muddied the waters by saying things like, oh, science hasn't proven that vaccines are safe, or science hasn't proven that they really don't cause
autism. You linked
autism,
that, right?
He did outright. And so I think that, putting that seed of doubt is problematic.
Completely.
Right? Because it's, just by saying like, well, we don't really know. It's like, no, we know. We know from hundreds of studies that have studied hundreds of thousands of children.
No, totally.
We do know.
We know. This is everything he says is driven to support his constituency because he wants to get reelected. Bottom line, nothing he says you can take at face value. I'm sorry.
And it's interesting, and that how different, because when it comes to vaccines and GMOs, two things that I am pro, it is interesting how the political lines, it doesn't always follow what you would anticipate. So what I've noticed, at least in my own circles, particularly my friends who are more conservative are actually pro-vaccine, but they've recently been they've been waning on that. They haven't really been as pro-vaccine as what I've seen in previous years. And I don't know if it's because of, what we're talking about here. So and also I think a lot of it too has to do with this idea of freedom, vaccine freedom. I think in TWIV you talked about it, Vincent.
Yeah.
But, unfortunately, something as simple as him coming out and saying that he was vaccinated and he supports it would go a long way. So,
of course, that's public health leadership. That's the whole point. Exactly.
Yep.
These 50,000 people who die of flu this season, most of them are not vaccinated. So if you want freedom to die, by all means, you can have that freedom. But, it's better if everyone's immune.
Right.
Hey, Immune, that's the name of our podcast.
Hey, Immune.
And you know what? You can find it at Apple Podcasts, microbe.tv/immune. Subscribe on your phone or tablet so you get every episode. Hey, it's only one a month. Get them all. And send your questions and comments. We have more, but we'll check them next time. Immune@microbe.tv. And next time, we'll have a book contest. How's that?
Oh, yeah.
I have a really cool book from ASM Press about immune cells, and we'll give it away next time. That'll be next month. If you really like what we do, consider supporting our shows. There are lots of them over at microbe.tv. You can go to microbe.tv/contribute. We have a couple of different ways you can support us. Cindy Leifer is at Cornell University in Ithaca, New York. Thanks, Cindy.
Thank you.
And you can find Cindy on Twitter. She is at Cindy Leifer, all one word. Steph Langel is at Ohio State University. Thank you, Steph.
Thank you. This was fun.
Stephanie Langel on Twitter. I'm Vincent Racaniello. You can find me at virology.ws or Prof VRR on Twitter. The music you hear on Immune is by Steve Neal. His work is at stevenealpercussion.com. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.